All right, let's dive right in. Today, we're going microscopic to look at a true masterclass in biological efficiency. It's a story all about survival, about saving energy, and about some of the most clever genetic programming you can possibly imagine.
And really, it all boils down to this one question. Bacteria live in a world that's constantly changing, and for them, wasting energy is just not an option. They have to make exactly what they need, only when they need it. Wasting precious resources to build something they could just get for free, that could literally be the difference between life and death.
And that, of course, brings us to the hero, or maybe the problem of our story, a little molecule called tryptophan. For a bacterium like E. coli, tryptophan creates this constant challenge that demands a really sophisticated solution.
So, what exactly is tryptophan? Well, it's an essential amino acid. Think of it like a very specific, absolutely crucial Lego brick. The cell needs this brick to build all the proteins that keep it alive and functioning. And it has two choices. It can either find these Lego bricks just floating around in its environment, or it can make them from scratch.
But, and this is a huge but, making tryptophan is incredibly expensive. It's a major drain on the cell's energy budget. It requires a whole assembly line of different enzymes, burning through resources. So if tryptophan is just freely available, making more would be like running a car factory at full capacity when the parking lot is already completely full of cars. It's just wasteful.
So you can see the dilemma here, right? If there's plenty of tryptophan around, the cell has to slam the brakes on production to save energy. But if tryptophan levels are low, it absolutely has to fire up that factory or it will starve. What the cell needs more than anything is a switch.
And guess what? It has one. Actually, it has two. Let's start with the first layer of control, a mechanism called repression. The easiest way to think about this is as the main on-off power switch for the entire tryptophan factory.
And the way this is all set up on the DNA is just brilliant. The 5 genes that actually build the tryptophan-making enzymes are all bundled together in this neat little package called the Tryp-operon. It's like a single blueprint for the whole assembly line. Right at the beginning of that blueprint is a stretch of DNA called the operator. That's our switch. And then somewhere else on the DNA, there's another gene that makes the protein that's going to flip that switch, the Tryp repressor.
So how does this work? Well, when the cell is low on tryptophan, it's go time. The repressor protein, by default, is in an inactive shape. It can't grab onto that operator switch. This leaves the genetic track wide open for the cell's reading machine, RNA polymerase, to come in and do its job. It reads the genes, the enzymes get made, and the tryptophan factory is up and running. But what happens when the cell has plenty of tryptophan? This is where it gets really elegant. The tryptophan molecule itself becomes part of the switch. It's what we call a co-repressor. It actually binds to that repressor protein, and when it does, the protein clicks into a new, active shape. And that new shape is perfect for grabbing onto the operator. It latches on, physically blocking the RNA polymerase, and shuts the whole operation down. Factory is off. Now you'd think, problem solved, right? An on-off switch seems pretty solid. But for an organism that has to manage its resources with such incredible precision, is a simple binary switch really good enough? Or is there maybe another layer of control we haven't seen yet? And that's where we meet the second control system, an absolutely beautiful mechanism called attenuation. If repression is the big on-off switch, you can think of attenuation as the super-sensitive fine-tuning dial. It allows for a much more graded, nuanced response. The whole idea behind attenuation is just wild. It doesn't actually stop transcription from starting, it lets it begin, but then it yanks the emergency brake before the process can finish. The copying process kicks off, but it gets cut short before the machinery even gets to the important enzyme-building genes. And the secret sauce for this whole mechanism is hiding in this little piece of messenger RNA at the very start of the operon, called the leader sequence. It's incredibly clever. First, it has these 2 genetic words or codons right next to each other that specifically call for tryptophan. This is the cell's molecular sensor. And second, this little piece of RNA can actually fold back on itself to form different 3D shapes. And this is where it all comes down to a choice. The mRNA can fold into one of 2 competing shapes. If one part of the strand pairs with another, it forms what we call an anti-terminator loop. That shape screams, "Go!" Transcription continues. But if a different pairing happens, it forms the dreaded terminator loop, and that shape is a hard stop. It slams on the brakes.
Everything depends on which one of these two loops gets to form.
So how on earth does the cell decide which loop to make? Well, get ready, because this is probably the most brilliant part of the entire system. The cell uses the process of making proteins, translation itself, to sense tryptophan levels and control transcription.
To get this, you have to remember one crucial thing about bacteria, something totally different from our own cells. For them, making the RNA copy, that's transcription, and reading that copy to build a protein, translation, they happen at the exact same time in the exact same place. A ribosome literally jumps onto the mRNA and starts building a protein while the mRNA is still being copied from the DNA. It's a perfectly timed dance. Okay, so let's walk through it. Scenario 1. The cell is starving for tryptophan. A ribosome jumps on the mRNA and starts reading. But, it gets to those two tryptophan codons and it has to pause. It stalls. Why? Because there's no tryptophan around to be delivered by the tRNA molecules. And by physically sitting there stalled on the tracks, the ribosome covers up region 1 of the mRNA. This frees up region 2 to pair with region 3, forming that go loop, the anti-terminator. And just like that, transcription continues full speed ahead.
Now let's flip the script. The cell is swimming in tryptophan. That ribosome has no problem finding what it needs, so it zips right past those tryptophan codons without even pausing, and it only stops when it hits a stop signal a little further down. But look where it stops. By sitting there, it blocks region 2. This leaves region 3 free to pair up with the newly made region 4, and click. The 3-4 terminator loop snaps into place, transcription is shut down, and the factory is closed with pinpoint precision.
Okay, let's take a step back and look at the big picture here. E. coli isn't just relying on one trick. It's got this two-tiered system, and both parts are working together for one single goal: absolute, hardcore efficiency. And this really breaks it down perfectly. You've got repression. That's your big, chunky on-off switch. It handles the major changes, right? High tryptophan versus low tryptophan. But then you have attenuation, the super sensitive fine-tuning dial. It gives the cell a much more graded response because it's sensing the real-time availability of tRNAs that are actually ready for protein synthesis. It's an even more accurate measure of the cell's immediate needs.
When you put these two together, what you get is a system that is just unbelievably robust and responsive. It can handle the big swings with the repressor switch and then make tiny, precise adjustments with the attenuation dial. It ensures that not a single molecule of energy is wasted. It is a stunning piece of molecular engineering.
And that just makes you wonder, right? This is just one system in one type of bacterium. What other incredible, ingenious little switches, dials, and feedback loops are running the show inside every living thing on this planet? It's a powerful reminder that even in the tiniest organisms, there's an entire universe of elegant design just waiting to be explored.